Serology is another method of identifying microorganisms. Acute infections can be diagnosed serologically by detecting pathogen-specific antibodies in the serum. The presence of specific immunoglobulin M antibody shortly after the onset of symptoms is often diagnostic. Alternatively, specific antibody titers can be measured early and again at 4 to 6 weeks after infection. A fourfold rise in the titer usually is considered diagnostic. Assays for serum antibodies are very useful for the diagnosis of viral hepatitis. Antibodies that are not pathogen specific are produced by patients with syphilis or infectious mononucleosis, and assays for these cross-reacting antibodies are used in diagnosis. Molecular diagnostics is another method. Nucleic acid amplification techniques such as polymerase chain reaction, PCR, and transcription-mediated amplification are used for diagnosis of gonorrhea, chlamydial infection, tuberculosis, herpes, encephalitis, and today COVID-19, as on the picture. Proteomics. Mass spectrometry can be used to identify microorganisms based on protein content and has been introduced into routine clinical laboratories. It has the advantage of rapid identification of the bacterial species, but is not useful for antibiotic sensitivity that still requires culture. The Centers for Disease Control and Prevention, CDC, has evaluated the danger microorganisms pose as weapons on the basis of the efficiency with which disease can be transmitted, how difficult the microorganisms are to produce and distribute, what can be done to defend against them, and the extent to which they are likely to alarm the public and produce widespread fear. Based on this criteria, the CDC has ranked bioweapons into 3 categories designated as A, B, and C. The agents in the highest risk category A can be readily disseminated or transmitted from person to person, typically cause diseases that carry a high mortality rate with potential for major public health impact, may cause pandemics leading to widespread panic and social disruption, and are likely to require special action for public health preparedness. For example, the smallpox virus is a category A agent because of its high transmissibility, case mortality rates of 30% or greater, and the lack of effective therapy. Smallpox readily spreads from person to person, mainly through respiratory secretions and by direct contact with virus in skin lesions. Since routine smallpox vaccination ended in the United States in 1972, immunity has waned, leaving the population highly susceptible. Concerns that smallpox could be used for bioterrorism have led to reinstitution of vaccination for some medical and military personnel. Category B agents are less easy to disseminate, cause disease associated with moderate morbidity but low mortality, and require specific diagnostic and disease surveillance. Many of these agents can be spread in food or water. Category C agents include emerging pathogens that could be engineered for mass dissemination because of ease of availability, production, and dissemination, the potential for high morbidity and mortality, and great impact on health. Microbes can enter the host through breaches in the skin by inhalation or ingestion, or by sexual transmission. The first defenses against infection are intact skin and mucosal surfaces, which provide physical barriers and produce antimicrobial substances. In general, respiratory, gastrointestinal, or genitourinary tract infections that occur in otherwise healthy individuals are caused by relatively virulent microorganisms that are capable of damaging or penetrating intact epithelial barriers. By contrast, most skin infections in healthy individuals are caused by less virulent organisms that breach the skin through damaged sites. The dense keratinized outer layer of skin is a natural barrier to infection. Furthermore, the low pH of the skin, less than 5.5, combined with the presence of fatty acids, inhibit the growth of microorganisms other than the normal bacteria and fungi. Included among these normal flora are potential opportunists such as Staphylococcus aureus and Candida albicans. Cutaneous infections are typically acquired by entry of microbes through breaks in the skin, including wounds or surgical incisions, like in cases of staphylococci, burns, as in cases of Pseudomonas aeruginosa, and diabetic and pressure-related foot sores in multibacterial infections. Intravenous catheters in hospitalized patients provide portals for local or systemic infection. Needle sticks can expose the recipient to infected blood and transmit HBV, HCV, or HIV. Some pathogens penetrate the skin via an insect or animal bite. Bites by fleas, ticks, mosquitoes, mites, and lice break the skin and transmit arboviruses that could become a cause for yellow fever or encephalitis, as well as bacteria and helminths. Animal bites can lead to infections with bacteria such as Pasteurella or viruses such as rabies. Gastrointestinal pathogens are transmitted by food or drink contaminated with fecal material. When hygiene fails, as may occur with natural disaster such as floods and earthquakes, diarrheal disease become rampant. Acidic gastric secretions are important defenses and are lethal for many gastrointestinal pathogens. Healthy volunteers do not become infected by Vibrio cholera unless they are fed 1011 organisms. By neutralizing the stomach, acid reduces the infectious dose by 10,000-fold. By contrast, some ingested agents such as Shigella and Giardia cysts are relatively resistant to gastric acid, so fewer than 100 organisms can cause illness. Other normal defenses within the gastrointestinal tract include the layer of viscous mucus covering the intestinal epithelium, lytic pancreatic enzymes and bile detergents, mucosal antimicrobial peptides called defensins, normal flora, and secreted immunoglobulin A antibodies. Immunoglobulin A antibodies are made by plasma cells located in mucosa-associated lymphoid tissue. These lymphoid aggregates are covered by a single layer of specialized epithelial cells called M cells, which are important for transport of antigens to mucosa-associated lymphoid tissue or mouth. Numerous gut pathogens use M cells to enter the host from the intestinal lumen, including poliovirus, enteropathic Escherichia coli, Vibrio cholera, Salmonella enterica, serotype typhi, and Shigella flexneri. Infection via the gastrointestinal tract occurs when local defenses are weakened or the organisms develop strategies to overcome these defenses. Host defenses are weakened by low gastric acidity, by antibiotics that alter the normal bacterial flora, or when there is stalled peristalsis or mechanical obstruction. Viruses that can enter the body through the intestinal tract, including hepatitis A or rotavirus, are those that lack envelopes because enveloped viruses are inactivated by bile and digestive enzymes. A large number of microorganisms, including viruses, bacteria, and fungi, are inhaled daily by every individual. In many cases, the microbes are inhaled in dust or aerosol particles. The distance these particles travel into the respiratory system is inversely proportional to their size. Large particles are trapped in the mucociliary blanket that lines the nose and the upper respiratory tract. Microorganisms trapped in the mucus secreted by goblet cells are transported by ciliary action to the back of the throat, where they are swallowed or coughed out. Particles smaller than 5 nanometers travel directly to the alveoli, where they are phagocytosed by alveolar macrophages or by neutrophils recruited to the lung by cytokines. Microorganisms that invade the normal healthy respiratory tract have developed specific mechanisms to overcome mucociliary defenses or to avoid destruction by alveolar macrophages. Some successful respiratory viruses evade these defenses by attacking to and entering epithelial cells in the lower respiratory tract and pharynx. Their attachment induces the host cell to engulf the virus, leading to viral entry and replication within the host cell. The urinary tract is almost always invaded from the exterior by way of the urethra. The regular flushing of the urinary tract with urine serves as defense against invading microorganisms. Urine in the bladder is normally sterile or contains only a small number of fastidious bacteria. However, successful pathogens adhere to the urinary epithelium, overcoming the host defense of regular flushing. Anatomy plays an important role in infection. Women have many more urinary tract infections than men because the distance between the urinary bladder and skin, in this case the length of the urethra, is 5 cm in women, in contrast with 20 cm in men. Obstruction of urinary flow, as in benign prostatic hyperplasia shown on the picture, or reflux, can compromise normal defenses and increase susceptibility to urinary tract infections. Urinary tract infections can spread further up from the bladder to the kidney and cause acute and chronic pyelonephritis. From puberty until menopause, the vagina is protected from pathogens by a low pH resulting from catabolism of glycogen in the normal epithelium by lactobacilli. Antibiotics can kill the lactobacilli, allowing overgrowth of yeast with resultant vaginal candidiasis. Some microorganisms proliferate locally at the site of initial infection, whereas others penetrate the epithelial barrier and spread to distant sites by way of the lymphatics, the blood, or nerves. In contrast to those that disseminate, pathogens that cause superficial infections stay confined to the lumen of hollow viscera or interact exclusively with epithelial cells. Microbes can spread within the body in several ways. Lysis and invasion. Some extracellular bacteria, fungi, and helminths secrete lytic enzymes that destroy tissue and allow direct invasion. For example, Staphylococcus aureus secretes hyaluronidase, which degrades the extracellular matrix between host cells. Invasive microbes initially follow tissue planes of least resistant and drain to regional lymphatics. Staphylococcus aureus may travel from a localized abscess to the draining lymph nodes. This can sometimes lead to bacteremia and spread to deep organs. The other path for spreading in the body is through blood and lymph. Microorganisms may be spread either in extracellular fluid or within host cells. Some viruses like poliovirus and the HBV, most bacteria and fungi, some protozoa, and all helminths are transported in the plasma. Leukocytes can carry herpes viruses, HIV, mycobacteria, leishmania, and toxoplasma. The parasites Plasmodium and Babesia are found within red cells. The third way of transmission is cell-to-cell transmission. Most viruses spread locally from cell to cell by replication and release of infection. variants, but others may propagate from cell to cell by causing fusion of host cells or by transport within nerves. Infectious agents establish infection and damage tissues by any of three mechanisms. They can contact or enter host cells and directly cause death of infected cells. It's shown on the picture. They can release toxins that kill cells at distance, release enzymes that degrade tissue components or damage blood vessels and cause ischemic necrosis. They can induce host immune responses that, although directed against invader, cause additional tissue damage. Therefore, the defensive responses of the host can be a mixed blessing, helping to overcome the infection, but also contributing to tissue damage. The consequences of blood-borne spread of pathogens vary widely depending on the virulence of the organism, the magnitude of the infection, the pattern of seeding, and host factors such as immune status. Sporadic bloodstream invasion by low virulence or non-virulent microbes is common but is quickly controlled by normal host defenses. By contrast, disseminated viremia, bacteremia, fungemia, and parasitemia by virulent pathogens poses a serious danger and manifests as fever, hypertension, and multiple other systemic signs and symptoms of sepsis. Massive bloodstream invasion by bacteria can be rapidly fatal even in previously healthy individuals. The major manifestations of infectious disease may appear at sites distant from the point of microbe entry. For example, varicella zoster and measles viruses enter through the airways but cause rashes in the skin. Poliovirus enters through the intestine but kills motor neurons to cause paralysis, as shown on the picture. There is a hypothetical scheme of poliovirus pathogenesis based on experimental findings in humans, monkeys, chimpanzees. Ingested virus initially replicates in the oropharyngeal and intestinal mucosa. Virus replication at these sites reaches the blood through the lymph nodes, resulting in a primary viremia. Invasion of virus into the central nervous system may occur either directly from the blood or by retrograde axonal transport when the virus enters the neuromuscular junction. It is believed that invasion of the brain or spinal cord must be preceded by viral multiplication in extraneural tissues, which leads to a sustained viremia. These extraneural tissues may include skeletal muscle and brown fat. Viruses spread most frequently by the fecal-oral route. Shedding of virus from the nasopharynx may lead to transmission of infection by the respiratory route, which occurs in developed countries with high standard. The other major manifestation is schistosoma hematobium that also penetrates the skin but localizes to the urinary bladder and causes cystitis. The rabies virus travels from the site of the bite by a rabid animal to the brain by retrograde transport in sensory neurons, where it then causes encephalitis and death. Transmission depends on the hardiness of the microbe. Some microbes can survive for extended period in dust, food, or water. Bacterial spores, protozoan cysts, and thick-shelled helminthes can survive in a cool and dry environment. Less hardy microorganisms must be quickly passed from person to person, often by direct contact. For transmission of disease, the mode of exit of a microorganism from the host body is as important as entry into it. Every fluid of tissue that is normally secreted, excreted, or shed is used by a microorganism to leave the host for transmission to the new victim. Skin flora such as Staphylococcus aureus and dermatophytes and fungi are shed in the desquamated skin. Some sexually transmitted pathogens are transmitted from genital skin lesions such as HCV and Treponema pallidum. Oral secretions. Viruses that replicate in the salivary glands and spread in saliva include mumps virus, cytomegalovirus, and rabies virus. Respiratory secretions. Virus and bacteria can be shed in respiratory secretions during talking, coughing, sneezing. Most respiratory pathogens, including influenza viruses, spread in large respiratory droplets which travel no more than 3 feet. However, a few organisms, including Mycobacterium tuberculosis and varicella zoster virus, are spread from the respiratory tract in small respiratory droplets or within dust particles that can travel long distances in the air. These properties determine the type of isolation precautions that are used to prevent the spread of infection. Stool. Organisms shed in stool include many pathogens that replicate in the lumen or epithelium of the gut, such as Shigella and rotavirus. Pathogens that replicate in the liver, like hepatitis A virus, and gallbladder, like Shigella and Enterica serotype typhi, enter the intestine in bile and are shed in stool. Blood. Pathogens spread via blood may be transmitted by invertebrate vectors, medical practices, or sharing of needles by intravenous drug abusers. Blood-borne parasites, including Plasmodium and arboviruses, are transmitted by biting insects. Urine. Urine is the usual mode of exodus from the human host for only a few organisms, including Schistosome hematobium, which grows in the veins of the blood and releases eggs, as on the picture, that reach the urine. Genital tract. Sexually transmitted infections spread from the urethra, vagina, cervix, rectum, or oral pharynx using genital tract. Organisms that cause sexually transmitted infections depend on direct contact for person-to-person spread because these pathogens cannot survive in the environment. Transmission of STIs often is asymptomatic by asymptomatic individuals who do not realize that they are infected. Infection with one sexually transmitted infection increases the risk for additional sexually transmitted infections, mainly because the risk factors are the same for all STIs. Vertical transmission. Transmission of infectious agents from the mother to fetus or newborn child is a common mode of transmission for some pathogens and may occur through several different routes. Placental fetal transmission is most likely to occur when the mother is infected with pathogen during pregnancy. Some of the resulting infection damage depend on the age of the fetus at the time of infection. For example, rubella infection during the first trimester can lead to heart malformations, mental retardation, cataracts, or deafness, while rubella infection during the third trimester has little effect. Congenital microcephaly and other central nervous system complications have been associated with Zika virus infection during pregnancy. Much is still unknown about timing of infection relative to the trimester of pregnancy. Transmission during birth is caused by contact with infectious agents during passage through the birth canal. Examples include gonococcal and chlamydial conjunctivitis. Postnatal transmission in maternal milk can transmit cytomegalovirus, HIV, and hepatitis B virus. Microbes also can be transmitted from animal to human, resulting in zoonotic infections either through direct contact with or consumption of animal products or indirectly by an intervertebrate sector. Viruses can directly damage host cells by entering them and replicating at the host expense. The manifestation of viral infection are largely determined by the tropism of the virus for specific tissues and cell types. Tropism is influenced by a number of factors: host receptors for viruses, specificity of transcription factors, and physical characteristics of tissues. Viruses are coated with surface proteins that bind with high specificity to particular host cell surface proteins. Entry of many viruses into cell commences with binding to normal host cell receptors. For example, HIV glycoprotein GP120 binds to CD4 and CXCR4 and CCR5 on T cells and macrophages. Host proteases may be needed to enable binding of virus to host cells. For instance, a host protease cleaves and activates the influenza virus hemagglutinin. Specificity of transcription factors is another important issue to discuss. The ability of the virus to replicate inside particular cell types depends on the presence of lineage-specific transcription factors that recognize viral enhancer and promoter elements. For example, the JC virus, which causes leukoencephalopathy, replicates only in oligodendroglia in the central nervous system because the promoter and enhancer DNA sequences regulating viral gene expression are active in glial cells but not in neurons or endothelial cells. Host environment and temperature can contribute to tissue tropism. For example, enteroviruses replicate in the intestine in part because they can resist inactivation by acids, bile, and digestive enzymes. Rhinoviruses infect cells only within the upper respiratory tract because they replicate optimally at the lower temperatures characteristic of the site. Once viruses are inside host cells, they can damage or kill the cells by a number of mechanisms: direct cytopathic effects, antiviral immune responses, viruses can kill cells by preventing synthesis of critical host macromolecules, by producing degradative enzymes and toxic proteins, or by inducing apoptosis. For example, poliovirus blocks synthesis of host proteins by inactivating cap-binding protein. Herpes simplex virus produces proteins that inhibit synthesis of cellular DNA and mRNA and other proteins that degrade host DNA. Viral replication also can trigger apoptosis of host cells by cell-intrinsic mechanisms, such as perturbations of the endoplasmic reticulum during virus assembly, which can activate caspases that mediate apoptosis. Viral proteins on the surface of host cells may be recognized by the immune system, and lymphocytes may attack virus-infected cells. Cytotoxic T lymphocytes are important for defense against viral infections, but cytotoxic T lymphocytes also can be responsible for tissue injury. Hepatitis B infection causes cytotoxic T lymphocyte-mediated destruction of infected hepatocytes, a normal response that is attempting to clear the infection. Transformation of infected cells is another important mechanism. Different oncogenic viruses like human papillomavirus, Epstein-Barr virus can accumulate cell growth and survival by a variety of mechanisms, including hijacking the control of cell cycle machinery, anti-apoptotic strategies, and insertional mutagenesis. in which the insertion of viral DNA into the host genome alters the expression of nearby host genes. Coronavirus disease 2019, which is zoonotic in origin and most commonly spread through respiratory droplets or aerosol transmission, has caused a big threat to mankind. The disease was originated in the seafood market on Wuhan, Hubei, China in early December 2019, with clinical presentations greatly resembling viral pneumonia. The disease has affected most of the countries in the period of 2 months itself. The outbreak of COVID-19 has been declared a pandemic by World Health Organization and presents a great challenge for the healthcare communities across the globe. The presently identified causative agent for COVID-19, severe acute respiratory syndrome coronavirus 2, has shared 82% genome sequence similar to previously identified SARS-CoV-1, which also originated in China in 2002. The structure of the enveloped single-stranded RNA virus consists of spike protein, which is mainly responsible for the pathogenesis in the human species. The spike protein of the virus, through its receptor binding domain, gets attached to a human cell surface receptor protein, angiotensin converting enzyme 2, ACE2, encoded by the ACE2 gene, followed by its priming through auxiliary protein TMPRSS2, transmembrane proteases serine 2, a cell surface protein that is expressed by epithelial cells of specific tissues, including those in the aerodigestive tract. ACE2, which acts as a viral host cell entry receptor, has ubiquitous distribution on the organs. Therefore, SARS-CoV-2 in severe cases causes a systemic disease with possible involvement of the kidneys, the heart and blood vessels, the liver, the pancreas, and also regulates alterations in circulating lymphocytes and the immune system. The maximum expression of the ACE2 receptors, according to the consensus dataset from the Human Protein Atlas, is found in small intestine, duodenum, and colon, followed by kidneys, testes, gallbladder, heart, thyroid gland, adipose tissue, rectum, and lungs. The histopathological features of COVID-19 closely resemble those seen in SARS and MERS. SARS, which is a type of pneumonia caused by the SARS coronavirus, is highly contagious and can affect multiple organs. The SARS outbreak in 2003 prompted extensive studies of its histopathological features, with the discovery that the disease predominantly attacks the lungs and the immune system. The main histopathological changes can be summarized as lung disease, damage to the immune organs, systemic vasculitis, and differences in systemic toxicity and secondary infections. Injury to the lungs results in clinical acute respiratory distress syndrome, which corresponds to diffuse alveolar damage histologically. MERS is caused by the Middle East respiratory syndrome coronavirus. The histopathologic features comprise 3 major patterns: diffuse alveolar damage, multiple organ microvasculitis, and lymphocyte infiltration and changes in immune organs. Like SARS-CoV and MERS-CoV, SARS-CoV-2 mainly attacks the lungs, causing diffuse alveolar damage, as shown on the picture, with edema and hyaline membrane formation, which is accommodated by macrophage and lymphocytic infiltration to varying degrees. These findings are common to viral pneumonias in general. However, ongoing histopathological studies are determining the specific characteristics with more certainty. Ultrastructural examination can reveal more details regarding the viral particles. Type 2 pneumocytes harbor numerous autophagosomes characterized by double membranes and presence of organelles in the cytoplasm, as shown on the picture. These autophagosomes, which contain viral aggregates, may also be present in tracheal epithelial cells and within the extracellular mucus in the tracheal human. Presence of virus particles has been shown by immunohistochemical staining using monoclonal antibody against SARS-CoV-2 nucleocapsid protein. After multiple rounds of discussion among basic science researchers, pathologists, and clinicians working on COVID-19 have presented various hypotheses on the pathogenesis of SARS-CoV-2. Hypotheses for the pathogenesis of these findings at biopsy or autopsy include disordered immune response to the virus with a hyperinflammatory state and abnormal coagulation, which may be secondary to the immune response or related directly to viral injury of endothelial cells. Ongoing scientific research is needed to further explore SARS-CoV-2 and its impact on the human body and, by extension, global public health. Because the World Health Organization declared the COVID-19 pandemic, all health personnel, including pathologists, have been urged in a common effort of solidarity in the different disciplines in which they work, with an emphasis on prioritization of activities at this time of global health emergency, therefore maintaining a high level and optimal response time for routine diagnostic activity while concurrently investigating the COVID-19 process. Postmortem histopathological findings and biopsies could play an essential role in understanding the pathophysiology of SARS-CoV-2 infection. Indeed, using appropriate biosafety precautions, the US CDC and the WHO called for action to perform full and detailed autopsies on patients who have suffered from SARS-CoV-2 infection leading to COVID-19. Finally, further research using fresh, frozen, and formalin-fixed tissues obtained from autopsy or biopsy is needed to better understand the tropism of SARS-CoV-2 and the extent of its effects on different organs and tissues. For example, the central nervous system deserves in-depth analysis to reveal the impact of the virus on the brain in correlation with neurological manifestations and complication. Immunohistochemistry, genomic, and PCR-based techniques performed on these issues will be relevant for such investigation in the nearest future. Thank you for your attention.